So welcome to our webinar on the new platform DIAGAST Live, and thank you for your registration. So we are waiting for around 100 people, so we still wait a few minutes, please. And a little reminder, you can use the chat at the right of the screen to ask all your questions to Mr. Le Pendu. He will answer you at the end of his presentation. So now it's time to start this webinar. So we can start now. Let's go. Good morning, Laurent. I'll let you introduce yourself and our expert, Mr. Le Pendu.
Yes, okay, thank you. Thank you, Anne, and good morning. Welcome everyone, and thank you very much for your registration to attend this webinar. My name is Laurent Soufflet. I'm Innovation Manager at DIAGAST and also in charge of technological watch in the field of immunohematology and related areas as well. That in order to update our scientific validity report and post-marketing surveillance of our reagents and products. First of all, it's a real pleasure for me and a big honor to introduce this very first webinar of scientific series of the DIAGAST Live platform. I hope that this webinar inaugurates a series of interesting scientific meetings and will really become a valuable appointment for participants. The months of pandemics we experienced or that we are still living for is very special for everyone, and whether in life or professionally, every day, and for some of us at the individual or family level. So when we began to organize this webinar, the question was, as a supplier of immunohematology reagent and system, how could we help to diffuse and spread the knowledge and help to fight against the virus? It was the beginning of the year that rests on many scientists' mind still today. From the early beginning, of course, with DIAGAST, we have followed the evolution of the knowledge of these parameters that could involve our blood groups and potentially our blood grouping products, CPU or pad grouping device, and we decide to contribute to it by using the time of this webinar, particularly on this subject, without any other objective and scientific interest. It's well known that certain group is correlated with different type of pathologies, and I can list them today in this session, of course. Likewise, our goal is not to enter in the field of the use of CCP or this kind of thing, even if the group are an important component of these concerns. Indeed, all these aspects have been discussed at the ISBT earlier in June. Our purpose as producer of grouping antibodies and transfusion-related reagents is to focus on the relationship between the COVID-19 and the blood group at a biochemical and population immunological level. In this way, we asked to Jacques Le Pendu, simply one of the most important key opinion leader in the field of the host-pathogen interaction, to expose his original vision and review of these recent events. Of course, it's not necessary to present Jacques to you because his contributions are numerous and very high quality. And this question is still controversial. It seems that a consensus is emerging from the debate. And as a specialist, Jacques Le Pendu and his co-author have greatly contributed to explain very early the possibilities of such interactions. His presentation will be appreciated by all those who wish to benefit from a high-level inventory of this fascinating question. And technically, question could be addressed or sent directly at the chat zone, as described by Anne, and we will make a synthesis of it if it's necessary. So welcome, Jacques. It's up to you.
So thank you to DIAGAST for this opportunity to give this update on a hot topic regarding blood type. Well, you certainly heard about relationships between AB or blood groups and COVID-19. You may also have heard that due to some inconsistencies in the data or to an apparent small effect, it's often considered of little importance. What I would like to do here is to review the available evidence, but rather than going through a critical assessment of every single publication on the topic, I would like to analyze the data in the light of the potential underlying mechanisms and see how the data fit with what one can expect according to those mechanisms. As you will see, hopefully, with that approach, most inconsistencies in the data set can be explained, and the ABO effect on SARS-CoV-2 transmission appears rather substantial. Well, let me start back here.
Thank you.
And let's first look at those studies that attempted to observe associations between ABO blood groups and the disease severity. There are not many such studies well designed to observe a potential effect. They're listed here. There are a few more. This was in December 2020 when I compiled all studies. There are a few more since then, but they don't change the general picture. The trend we see is that blood group O seems to be somewhat protective in comparison with non-blood group O, and this is written with a lower risk in green. So severely ill blood group O patients tend to heal faster or have a decreased risk of death. Nonetheless, the observed effects were generally weak. There is one exception only from an Italian studies. It concerns hypertensive patients. In that case, it's a subgroup of patients, of high-risk patients. The relative risk of death from non-O patients was over 3 times higher as that of blood group O patients. So being a non-O patient is a really high risk factor. So I'm aware of only one study specifically concerned with these high-risk patients. It would certainly be interesting to conduct more studies to validate or invalidate the observation, as it may have practical consequences on patients' care. Additionally, I would like to mention the results of a very impressive study recently reported by Patak and colleagues. In their work, the authors performed a multi-level transcriptome-wide association study of genetically regulated expression by integrating splicing and proteome expression with genome-wide summary statistics on COVID-19 hospitalization on a large number of patients, as you can see here. That allowed identification of a small subset of genes associated with COVID-19 hospitalization. Only 2 genes, OAS1 and ABO, significantly came out. I'm sorry, with all the changes we made, I forgot to turn on the pointer. So they came out with these various genes, and only 2 of them, OS1 and ABO, significantly came out in the 3 approaches. So following this pre-selection, the authors performed a gene-based PHIWAS, or a phenotype-wide association study. And they observed that the ABO gene associated with patients developing acute pulmonary heart disease, pulmonary embolism and infarction, venous embolism and thrombosis, deep vein thrombosis, and pulmonary heart disease. In other words, ABO was clearly implicated in thrombotic and coagulation-regulated events. So the obvious question now is, how can we explain this association between the severity of COVID-19 and ABO blood groups? So the first answer comes from a large set of previous studies linking ABO blood types with thrombosis and the vascular function. Disorders of hemostasis are a critical aspect in the severity of COVID-19. So the lower risk of thrombosis in people of blood group O likely explains their better prognosis under intensive care. So, you can see here blood group O is associated with a decreased risk relative to non-O of several thrombotic-associated diseases. This is due to decreased plasma levels of coagulation factors, and in particular, von Willebrand factor. But also there are effects on the vascular function underlined by decreased plasma level of ICAM, P-selectin, and E-selectin, therefore adhesion molecules. But the detailed mechanisms are not well known so far. A second type of potential explanations for these associations between ABO and the severity of COVID-19 might be related with the fact that ABO types turn out to be associated with comorbidity factors such as inflammatory disease and metabolic disorders. Again, with blood group O appearing as a protective factor, but the mechanisms so far are entirely unknown, potentially involving the microbiota, though. Yet most published studies reporting relationships between ABO blood types and COVID-19 have been concerned with the risk of being ill or infected and not with the disease severity. So let's take a look at those studies now. This table here gives a quick summary of studies available by the end of 2020 that I compiled. There have been a few more since then, but they don't change the general picture, so I did not include them to keep the table readable. What is apparent here is that most studies reported a lower risk of people for people of blood group O in comparison with non-O, and this is boxed in green here, or inversely a higher risk of non-blood group O individuals, in particular blood group A, and that's boxed in red. This does not seem to depend on the number of cases enrolled, which is highly variable, or on whether the studies significantly or specifically, looked at the ABO gene or included whole genome analysis. With these studies of GWAS studies are marked here in red. Studies came from different parts of the world, and we will see later that it matters quite a lot. In addition, I should mention that several studies at the bottom of the table here failed to find any significant association between blood group O and COVID-19. So overall, blood group O appears associated with a decreased risk of COVID-19, this has been more detailed through a meta-analysis performed by Franchini and colleagues. And you can see on this table here, on the associated figure, that it generated a significant association where the risk for blood group O individuals is decreased by around 20%. But again, how can we explain these results? The first and most straightforward explanation is that some blood types would be intrinsically more susceptible to infection than others. And this has been observed previously for other viruses. In the case of SARS-CoV-2, the same explanation has been proposed by some researchers who reported that the RBD, the receptor binding domain of the spike protein from SARS-CoV-2, would attach to blood group A type 1, which is shown here. That is a particular structure of the A antigen expressed on lung or digestive epithelial cells. No binding was observed to A type 2 that is expressed on red cells. The two structures are constituted by a tetrasaccharide and differ only by the linkage to the underlying N-acetylglucosamine, which is in beta 3 here in the case of A type 1 and in beta 4 in the case of A type 2. So Wu and colleague, this American team, observed binding of the RBD only to A type 1. But it should be kept in mind that if it was a major mechanism, one should observe a higher impact of the epidemics in regions where blood group A is most frequent. And that is not the case, as we will see later. So we tried to reproduce these observations. For this purpose, we produced a recombinant RBD protein fused to the Fc domain of a mouse immunoglobulin. An incubation of this fusion protein to ACE2 shows a very strong recognition, as you can see here, as expected. However, we could not detect any binding to either A type 1 or the underlying precursor H type 1 at a fairly high concentration. So we also tried to see if this fusion protein could bind to salivary mucin from secretory individuals of the A, B, or O type, and we did not detect any binding above the background level. Likewise, we tried to see if there was any binding to the blood group A epitope present in the lung, on the lung tissue of a blood group A individual. You can see that staining of the tissue by an anti-A antibody is very strong, and yet the RBD Fc protein, fusion protein, does not detect anything similar to what is observed is only counter-staining on blood group O.
Excuse me, Jack. I have another question from another channel. Are other viruses able to produce attachment proteins recognizing A antigens in such model?
Oh, yeah, it is known. This has been observed previously for two types of viruses, noroviruses and rotaviruses.
Okay.
But not for every single strain of norovirus or every single strain of rotavirus. It is very much strain dependent.
Okay.
So depending on the strains, different blood types are recognized. And a specific type is only seen in the case of several rotavirus strains that are of zoonotic origin. Okay, so, well, at this stage, we were therefore unable to reproduce the results, suggesting that people with blood group A would be more susceptible to infection by SARS-CoV-2 due to recognition of the antigen by the virus attachment protein. So we have to turn ourselves to another type of explanation. And the second type of explanation for these associations between ABO phenotypes and COVID-19 is related to context-dependent susceptibility based on the presence of natural anti-carbohydrate antibodies. We know that human plasma contains a large repertoire of anti-carbohydrate antibodies. Most targets of these antibodies are bacterial polysaccharides or LPS, but also some mammalian glycans are strongly represented. First off, the xenoantigens, alpha-gal, forceman, or the glycolyl form of salic acid called NuGC. Also, there are cryptic or truncated motifs such as the TN, T, TK, the O-glycan core 3 or 4, Lewis C, polylactosamine structures terminated by N-acetylglucosamine or the PK antigen, and very interestingly, the alloantigens ABO of the ABO system. And so it's been shown earlier that animal enveloped viruses which carry the alpha-gal antigen, the alpha-gal antigen is a xenoantigen not present in humans because we have lost the gene to encoding the alpha-gal transferase required for its synthesis, but it's very closely related to the B blood group antigen. And so it's been shown that anti-alpha-gal antibodies neutralize a series of viruses marked in red here. So such anti-carbohydrate antibodies do neutralize viruses both in vitro and in vivo. But also it's been shown in vitro that anti-A or anti-B blood group antibodies neutralize measles virus or also HIV produced in cells expressing the A or the B antigens. Similarly, anti-A antibodies can block the interaction between ACE2 and the SARS-CoV S protein from 2002-2003, providing this S protein was synthesized by cells expressing the A blood group antigen. So we have this precedent. Now coming to SARS-CoV-2, we can see on this picture that I borrowed from a very nice molecular modeling study that the surface of its spike protein is largely covered by glycans, shown in dark blue here, that hide the peptide part, which is only visible in light blue or turquoise. This sugar coat provides what is called a glycan shield, since glycans are produced by the host cell itself and not encoded by the virus genome. Therefore, they are not immunogenic unless they constitute either cryptic xenoantigens or alloantigens, such as those of the A and B, the AB antigens of the ABO system. However, no characterization of glycans from authentic variants from patients is available so far. Nonetheless, one may expect that A and B blood group antigens, or the precursors, their precursor, called the H antigen present in blood group O people, to be present on the viral envelope proteins. Since these are synthesized according to the individual's blood group by the cells where the virus replicate. And again, you can see this picture showing a strong expression of the A blood group antigen on the tissue from a blood group O person. Of course, it's absent in a blood group from a blood group A person. Sorry, it's absent from a blood group O person. The antigen is expressed on the bronchi cells, also the alveolar cells, and the vascular endothelium. So the antigen is expressed by cells that replicate the virus. So to document the presence of these ABH blood group antigens on the spike protein of the virus, what we did was to transfect cells that do not express these antigens, and the negative controls are shown here. First with an alpha-2 fucosyltransferase, so we can generate expression of the H epitope at the surface of the cells, and we further transfect them with the A blood group enzyme or the B blood group enzyme, and we obtain cells that express these antigens. Now, if we transfect them further with the cDNA coding for the S1 domain of the spike protein, well, we obtain a spike protein that expresses either the A antigen when produced by cells expressing the A antigen, or the B antigen when produced by cells expressing the B antigen. So the viral spike protein can be tagged with the A, B, or H antigens according to the ABO phenotype of the cells that produce it. So now I would like to spend some time on the consequences this could have on the transmission of SARS-CoV-2, as it is not straightforward. If we make the hypothesis that natural anti-ABO antibodies can neutralize the virus, here is how this could affect transmission. Let's consider a blood group A individual releasing a blood group A antigen tagged variants, marked in, depicted here with red spikes. When encountering a blood group O person, the virus might be neutralized by the presence of the anti-A antibodies. And that is shown by the dashed arrow. Inversely, if an O blood group individual releases variants and encounter an A blood group individual, there is no such protection possible, and therefore this is marked by a plain arrow. So overall, what we have here is a model that corresponds to the basic rule of blood transfusion. The universal donors, blood group O people, and universal recipients, blood group AB people, since they are devoid of anti-A and anti-B. This generates several important consequences that can be tested on real-life data. First, the more frequent the blood group O in a population, the less protective it should appear in epidemiological studies. This is because blood group O individuals cannot be protected from transmission by other blood group O people, as illustrated by the circular arrow. By contrast, where blood group O is rare, these people should mainly encounter incompatible individuals and therefore benefit from a good protection by their natural anti-A and anti-B antibodies. And we can see that the number of incompatible ABO encounters will depend on the relative frequencies of the ABO phenotype. We know these are quite variable across geographical areas. So the model predicts that in areas where A, B, and O phenotype are well balanced, transmission at the population level should be optimally decreased in comparison with areas where A, B, and O frequencies are largely imbalanced. Also, one expects that anti-A and anti-B titers should matter. We know that the levels of these antibodies are greatly variable between individuals of the same blood type. Therefore, one may expect that people with high levels of these natural antibodies should be less likely to be infected. All this has been modeled either for the SARS-CoV-1 outbreaks from 2002, 2003, and more recently for the contemporary SARS-CoV-2 epidemics. This theoretical modeling indicates that if all incompatible ABO transmission were neutralized, then the epidemic would be considerably slowed, as you can see here, in comparison with the situation where anti-ABO antibodies would not play any role. So now let's see if any of these predictions holds up with real-world data. Well, the various studies that I mentioned before originate from various parts of the world. It is therefore possible to compare them according to the frequency of blood group O in the corresponding geographical areas. As predicted by the model, the risk of being infected for blood group O individuals appear lower, as you can see here, in areas where their frequency is below 40%, in comparison with areas where their frequency is over 40%. Also, that's the first that was the first prediction of the model that is fulfilled. The model also predicts that the epidemic impact should be more or less important depending on the relative frequencies of the ABO phenotype. In areas where these frequencies are such that the number of incompatible encounters are high, the attack rates should be lower than where they're low. Typically, in populations where A, B, and O frequencies are balanced, such as in some Asian countries, as this is marked in orange here, the calculated theoretical degree of R0 suppression should be maximal. Whereas in areas of high imbalance, between where blood group O is highly prevalent, for example, such as in Latin America, the theoretical degree of R0 suppression should be lower. And what we see here is a plot of the theoretical degree of R0 suppression against the infection attack rate as determined by seroprevalence data from September 2020. And you can see that there is a very strong relationship between these two parameters. Even if we look at the European level, where the ABO frequencies do not differ so much from between the various countries in Europe, the relationship is still visible. So these data are very strongly in favor of a frequency-dependent mechanism that functions in accordance with the rules of blood transfusion. Now, another prediction of the model, as I mentioned earlier, is that natural anti-A or anti-B antibodies should matter. So it's well known that these are highly variable. And you can see here the distribution in red of anti-A or anti-B antibodies in healthy controls. And you can see people with high levels and people with very low levels of these antibodies. And now if we look at patients who were recently infected, they tend to have lower antibody titers. That was quite significant. no difference, by contrast, was between controls and patients was observed for several other anti-carbohydrate antibodies, T, TK, Louis C, PK, glucnac, the alpha-gal, and the Forceman xenoantigens. So indicating that the difference between patients and controls for the anti-ABO antibodies was not due to a general decrease in anti-carbohydrate antibodies following infection. It was really specific for the anti-A or anti-B antibodies. So far, the lines of evidence that I mentioned in favor of the role of natural anti-A blood group antibodies in conferring some protection from SARS-CoV-2 infection have been indirect. So to determine whether it would be possible to obtain a more direct evidence, we undertook a study where one could analyze the outcome of ABO incompatibility in a situation of high-risk transmission. So we studied symptomatic COVID-19 hospital personnel and their spouses with known ABO blood groups. We could obtain 333 couples who met our inclusion criteria. All of them had to share the same bedroom, all couples. One member of the couple became ill and the other member either followed or not, no later than 8 days after the first one, and all infections were RT-PCR validated. We could therefore know the direction of transmission whenever it occurred. The ABO frequencies of in our cohort of 666 people were similar to those given by the National Blood Transfusion Institution in France. And the rate of transmission between spouses was 39%, which is similar to the 38% rate between spouses that had been calculated in a meta-analysis of 44 studies recently reported. So here one can compare the couples where transmission occurs in an ABO-compatible manner with those where transmission occurred in an ABO-incompatible manner. The figure shows the number of couples where transmission occurred under compatibility or incompatibility. And transmission cases are shown in black, whilst transmission, a lack of transmission between within the couple are shown in white. And so one can clearly see here that the proportion of transmission is very much decreased in case of incompatibility. The decrease is over 40%, indicating directly that ABO incompatibility is protecting from SARS-CoV-2 infection. Importantly, attack rates did not differ between ABO blood types, but only by the compatibility situation. Even blood group A were less frequently infected in the case of incompatibility than in the case of compatibility. So that confirms the potential that here there is no intrinsic resistance of blood group O individuals, as they also are quite highly susceptible in the case of compatibility in comparison to incompatibility. So intrinsic factors are unlikely to play a significant role in transmission. So before concluding, I would like to discuss why in the epidemiological studies that I presented earlier, blood group A was most often an apparent risk factor, whilst blood group O was an apparent protective factor, and blood group B generally yielded non-significant results. Well, one should bear in mind that most studies originated from areas where people of European descent are a majority. The ABO frequencies in these countries are such that blood group A people may only benefit from an incompatible transmission somewhere between 11 and 50% of encounters, 13% precisely in France, with an infected person. Whereas people of blood group B and O would benefit from a much higher rate of encounter with incompatible individuals, 47 and 55% respectively in France. So now blood group B seldom shows up in these population-based studies. The reason is that because most often there are too few to generate significant data, and likewise the same holds true for blood group AB, of course. So therefore, one can conclude that blood group O individuals are not intrinsically less susceptible, or inversely, that blood group A people as well as B and AB people are not more susceptible to COVID-19, to SARS-CoV-2 infection or COVID-19. It all comes down to a matter of chance encounter with compatible or incompatible infected individuals. And to finish off, I would like to examine if one can now explain why some studies failed to find a link between ABO blood types and resolve and COVID-19 and resolve the controversies that followed. First off, several studies were underpowered when considering the ABO frequencies in the concerned population. Also, there have been studies where the population heterogeneity was not taken into account, erasing the potential effect. Also, when considering studies where blood group O has a too high frequency, then you now, I hope, understood that the effect is largely erased because blood group O people will most often encounter other blood group O people, therefore in a compatible situation. Other studies compared non-infected controls to intensive care patients, who fortunately represent a small subset of all patients only, but with strong comorbidity factors that obscure the effect of ABO blood groups. Finally, there are studies that were conducted too late during the outbreak. Typically, a cohort study performed on a French aircraft carrier where 76% of sailors were infected due to complete absence of preventative measures. If we come back to the mathematical modeling, one can predict that the ABO effect should be more or less visible depending on the time since the start of the epidemic. Indeed, the most susceptible individuals, blood group A individuals, blood group AB individuals, will be infected first because they do not benefit from any protection by anti-A or anti-B antibodies. So they acquire specific immunity. Therefore, their frequency will decrease in the newly infected cases. By contrast, one should see now the proportion of the protected individual, the most protected individuals, blood group O individuals, becomes high as the epidemic unfolds. In other words, when too many people have already been infected, the effect apparently vanishes. This might also explain why two recent studies from the US failed to find any effect, even though a large number of cases and controls were analyzed in those studies. So to conclude, I believe that one can now safely state that blood group O is associated with a better prognosis when severe COVID-19 is present. Although the effect is generally small, it can be quite high in patients with a history of high blood pressure, who may thus require special attention. The mechanism is most likely involving effects on hemostasis, potentially cell adhesion and the microbiota, but a lot of work remains to be done to fully understand these mechanisms. Also, and completely independently, the risk of transmission is decreased for blood group O individuals who therefore benefit from an apparent protection. There is no intrinsic resistance or susceptibility depending on the ABO blood groups. But there's a strong ABO incompatibility effect, which is frequency dependent and leads to a decrease in the coefficient transmission, R0, that slows the epidemics. And that is quite similar and quite equivalent to a social distancing effect. Finally, and to finish off, really, I want to ask you a question. Should we try to raise the anti-ABO titers at the population level? And if yes, how should we best perform that? And with this, I would like to thank my colleagues in my team in Nantes, Nathalie Ruvren, Chloé, Adrien Bremont, and Josabelle Rocher, who performed a lot of work on the topic, and also our Belgian colleagues from the Hospital Brugman in Brussels, Anna Nelkens and Marie Delers, for the mathematical modeling and statistical analysis in Nantes, Véronique Sebi, and also in Nantes from our hospital, Rachida Boukari. And thank you for attention.
Okay, thank you very much, Jacques, for the great presentation. We can start the question session now, if possible. But I have just one question before, if you want, about other systems like Lewis P1 or even in the secretor status. Are they involved for the spread of infection at the same level? What is the role of compound antigens like A, Lewis B, B, Lewis B, and so on?
Well, I haven't seen any study on those antigens actually. Okay. They might be present on the spike protein as well, as it is synthesized by cells that have the enzymes to synthesize those antigens. But the anti-Lewis antibodies are not so frequent, as you know well, and so maybe the importance will be much less than that of the ABO antigens.
Okay.
But there is no data.
Yeah. Okay. Do you imagine that the other kind of what are the potential application of this vision of this model? Is it possible, for example, to try to quantify the immune response by antibody detection and quantification, or mucosal detection of the antibodies for diagnostic or control, like IgA or some? Or
yes, I think this is a very interesting question. I believe it might be interesting to look for, as you mentioned, mucosal antibodies, anti-A and anti-B antibodies, and possibly IgAs in particular. This has not been studied so far, and maybe they relate much better to the protection effect. This would be very interesting to study. I think we have seen, as I presented, some relationship between the plasma titers of agglutinating actually and anti-A or anti-B. So this is mostly IgM, plasmatic IgM, but it would be really interesting to look at anti-A antibody in the mucosa, and also focusing maybe on the IgAs.
Okay. Okay. Has anybody have a question about the subject? No question for the moment, so I have the time to ask another question. For your point of view, do you know that what about the IgG response could have a facilitating effect on infection, as we've seen for other kind of viral infection like HIV or so on?
Oh yeah, this is also an important point. Thank you for raising it. Well, we have no data so far. So really, something that is missing is to determine in vitro the ability of anti-A or anti-B antibodies, regardless of their isotypes, whether they are able to neutralize in vitro or, as you mentioned, to facilitate infection. That has not been done so far. And I'm aware of undergoing studies on this, but I am not aware of solid data so far. But nonetheless, I would like to be optimistic in the sense that our observation on the couples that I reported show that certainly there is no facilitating effect overall, because when the transmission is under incompatibility, the risk is much decreased, almost by half. So we do not see any facilitation effect there. We do see a protection effect.
Okay. Another question: do you think that it's better from your population approach to have a particular attention to A/B groups in order for vaccination?
I am not completely sure to understand your question. I'm sorry.
You think that the first effects are very thin, depending on the population rate of the different groups. Like the A/B are more exposed or have more infection during the beginning of the epidemic.
Yes.
Do you think that it's better for population protection to have a particular intention of vaccination of the A/B groups?
Oh, you mean, okay. Well, I think it might be a good idea. They do not benefit at all from any ABO incompatibility effect, so they clearly are at a higher risk. In most studies reported so far, they appear somewhat with a higher odd ratios of being infected, but it hardly reaches statistical significance because there are too few. But certainly they are at a higher risk. So yes, they do constitute a higher risk population. They might benefit from vaccination with some priority.
Okay, could be an idea. Okay, so we have no more questions for the moment. I don't know if we have our time enough.
We have time. We can, if you have another question, it's still time to ask. If not, we can close the webinar.
Okay. So if you have, did you have any publication or reference paper you could suggest a part of your own paper, of course, in order to go from?
Yes, I could suggest a couple of papers that seem particularly interesting, which I'm quite willing to share.
Okay.
Just need to have a link to do this, but yes, certainly. With pleasure.
To participant. So we have to thank you. Thank you a lot for your presentation and for our attendees. See you at the next webinar.
Okay, thank you, Laurent.
Well,
thank you, Mr.
Jacques.
Thank you, Mr. Le Pendu.
Goodbye.
Thank you, everybody.